cancer diagnosis at the age of 32, my mom and I talked a lot about the shock of that diagnosis, given several doctors told us you're too young to have endometrial cancer. After much discussion, we began to feel driven to inform and help educate other young women about what symptoms they should be concerned.
Having both been teachers, it seemed natural for us to look for a way to share Alex's cancer journey in order to help other young women avoid such a shocking diagnosis or possibly navigate a similar path. A podcast seemed the best way to get the word out, and that's how Down There Aware was born.
4 years later, our mother-daughter duo has gone from educating and informing to advocacy and action, with a fresh and sometimes very interesting and somewhat humorous multi-generational perspective.
We look forward to sharing with you Season 4 as we advocate, take action, and make it a priority to always be Down There Aware.
Well, thanks for joining us today. Today we have two really special guests from a company called Travera, who has some really awesome cancer technology that we want to bring to you. So we have Cliff and Dennis, and so we're going to let them introduce themselves because they know who they are best.
So hi, I'm Cliff Reed. I'm the founding CEO of Travera. We spun Travera out of MIT in 2018 to commercialize a fabulous new technology invented at MIT. And we'll go through that new technology a little later today. Prior to Travera, I was the founding CEO of Complete Genomics, which is one of the leading DNA sequencing companies. So I come to the field of cancer first through the lens of DNA and what we can do in genomic medicine, and now through a completely different approach to finding the right therapies for the right patient at the right time. Dennis?
Thanks, Cliff. So hi, my name is Dennis Watson. I'm the Vice President of Business Development with Travera, and I've been here right out of here. It has been a fast year, but very excited to be here and to be working with this amazing technology that the team has developed. But my background has been in the genetics and genomics market in cancer for about the last 15 years or so. So worked with two fairly large companies the last 5 years of that, really leading kind of the U.S. commercial organization for a breast cancer diagnostic assay. But the majority of that past 15 years has been in direct physician education and sort of bringing those products to market in the genetic and genomic environment. And so I'm well versed in diagnostics and the day-to-day of clinical practice. And so this is what I'm trying to help the team here do, is really bring this to the forefront that way.
That's incredible. Your work is super important. So we thank you, and we thank you for connecting with us. And I really think it's a great message to a lot of our listeners.
Excellent.
So the technology you mentioned came from MIT. Can you speak to a little bit about what is that technology? And, clearly it was something that took off and has really advanced. So just tell us a little bit more about that.
Yeah, and let me put it in the context of genomics, because so much of precision oncology now is based on DNA sequencing, and Dennis and I both come from that world. So the big challenge with genomics is it just doesn't reach enough patients.
Right.
When it's good, it's great. Drugs like Gleevec for CML and drugs like osimertinib for non-small cell. I mean, these are really important and powerful drugs enabled by the genomics revolution. But if you look across, all cancers, and also look just into ovarian cancer, there's very little that actually affects most cancer patients. Only about 10% of cancer patients get any benefit at all from all of genomic medicine. So I noticed that was sort of the trend in genomic medicine, and left my DNA sequencing company and ended up meeting up with a professor at MIT who'd invented an extraordinary new measurement tool. And what this new measurement tool can do is it can weigh single cells. Now, it sounds kind of simple and obvious. How does this relate to cancer? But it is an amazing tool because it weighs single cells with extraordinary accuracy. In fact, it weighed him to an accuracy. It was so small that we could detect the change in the diameter of a cell of about 5 nanometers.
Wow.
Remember, the wavelength of visible light is like 500 nanometers.
Right.
And everybody who weighs cells uses a microscope to do it. And so there's no way that they're going to ever be able to do anything as accurately as we can do. So we have this really cool new measurement tool for weighing single cells. But then MIT and Dana-Farber partnered over the last decade to see if this tool could be used to solve an age-old problem in cancer medicine. And that was, can we create for cancer a test just like works so beautifully well in infectious disease? It is, take the disease cells out of the patient, test a whole bunch of different drugs against those disease cells, pick the one that works and give it to the patient, right? This works for like 99% of all patients with a bacterial infection, and it's never worked in cancer. So with this ability to measure this tiny, tiny little weight changes, MIT and Dana-Farber said, let's run the experiment, take cancer cells out of a patient, apply a cancer drug to the patient's cells, and then weigh the cells and see if the cells change their weight in response to an effective cancer drug. And it turns out they do. And not only do they do in the first cancer that was tested at MIT, which is multiple myeloma, apparently they do across virtually all cancers. Wow. And virtually all cancer drugs. So we at Travera took this amazing invention out of MIT, created the company Travera to commercialize it. And then just last year, we commercially launched the test so that we can take live cancer cells from a patient, test a panel of 20 cancer drugs against their live cancer cells, and based on this exquisitely accurate weight measurement technology, determine which drugs are going to work and not work for that particular patient.
That seems like an incredible skip the line, right? Like where instead of having all of these, well, yeah, you could try this and then we could do this and then we could do this. It's like, actually, let's do this little thing and then we'll just tell you what the answer is. It's kind of like a cheat code.
It's a cheat code. Yeah, we like that a lot. We call this a simple physics solution to an almost impossible biology problem. But the situation facing late stage patients where they need this cheat code is, and not just ovarian, across virtually all cancers, is that by the time a patient has reached a stage 4, first they failed all the fancy drugs. Right. All the frontline drugs that are really highly effective. They've seen those drugs and they're no longer effective. And what they're faced with is a very long list of very low effectiveness drugs. And the thing that we've learned is that that list of drugs may be 10 or 20 or 30 drugs long. In most circumstances, there's some hidden gems in that list. There are a handful of drugs in that list that are actually going to work really well for that particular patient. It's just that there's no way to know which they are. And so, obviously the only thing to be done in the absence of that information is trial and error. And, and some oncologists will, pick their favorite or pick the one that worked last time and one after another kind of walk through those drugs. But for many late stage patients, they don't have time to go through all of those drugs. So we're the cheat code that enables them to take that long list of candidate drugs that typically the response rates of those late stage drugs are around 20%, but there are a few hidden gems in there. And by running the test in our laboratory against that patient's live cancer cells, we can find those hidden gems and jump directly to those drugs that are going to actually help the patient.
Well, you said you had launched commercially. What exactly does that mean? I mean, how widespread is it, and who knows about it?
So it's not very widespread. We launched it in November, so we're about 6 months in, and mostly we're running clinical studies to continue to build up additional clinical validation evidence. But we found that patients started reaching out to us, hearing about us through the technical literature or through a hospital that was running one of our studies. And so what we decided to do was make it available through a CLIA certified lab. We went through the entire CLIA certification process so that a patient could, through their oncologist, get access to our technology directly without having to join a clinical trial, which might be very inconvenient for the patient. And what we are doing now, and I'll let Dennis expand on this because this is really his program, is that we have entered into a series of partnerships with disease foundations who have groups of patients who come together for support and education. And we are offering this test through these disease foundations for free. In return for the patients giving us their outcomes data.
Sure.
A few months later when they know whether or not a drug has actually worked for them. So we have essentially we're partnering with patients so that they get access to our test for free, which may inform their oncologist of a hidden gem of a drug to try. And we get more data that shows the predictive accuracy of our test. And we'll describe that data in a few minutes. But I want to turn that over to Dennis to talk about the commercial availability of the test.
Yeah, no, absolutely. So, we're working with physicians and cancer centers and other specialized laboratories all over the country in a lot of different respects. But the reality is that most physicians that hear about this test through their patient have probably not had any experience with it. And so, that's a big piece of what we're doing is we're working with those patients and getting connected to those physicians to help them understand what this is and what it isn't. It is an early test and it's still in development in a lot of ways. And and some physicians understandably are more apprehensive to try something that's as new as this. But as Cliff pointed out, what we're effectively doing is we're getting cells and sample from a patient, and we can talk in a moment how we do that. But we get their cells, we then get them into our lab, and we run as many as 20 different drugs on that specimen against those live cells, and then 2 days later send the report back to the physician. The drugs that we're running, for the vast majority of cases, are all FDA cleared drugs indicated for that disease. So, the doctor isn't going to get a report back of some completely crazy drugs that they would never consider giving.
Sure.
That's not really what the value proposition here is for the patient. The value proposition is what Cliff and Alex already kind of spoke to. When you get to that third line of therapy or that fourth line of therapy, the guidance that exists in the guidelines is more limited. It doesn't tell you what drug should come next after the first two or so.
Yeah.
It just says, here's 15 other drugs you can try or 20 other drugs you can try with. And, oftentimes the drugs are listed alphabetically. There's not a great deal of guidance. And so, there's a lot of other things that are indicated, but to Cliff's earlier point, identifying which of those things or which of those drugs are going to be best for this patient is the challenge. And it's not just a timing problem, right? It's not just about trying this drug and seeing if it works and then trying the next drug and seeing if it works and, and so on. I personally lost my mother-in-law to ovarian cancer and I've watched the battle closely. And every new drug you try is just, really breaks you down. It breaks you down emotionally. It breaks you down mentally. It breaks you down physically. And then the idea of then trying that next drug, you're in a more weakened condition, and it becomes sort of a snowball rolling down a hill, and not to mention the sort of mental toll of the failures. And so if we can take that list of here's the 15 drugs that are for late stage third line use, for example, and let's just better select which one comes next. Beautiful. And to the point of how we've been working with foundations and and the way we actually found Down There Where is, as Cliff mentioned, patients started finding us. They started talking about us, and there started to be a little bit of a groundswell. But this is early. The data is early. We're still going through a lot of validations and a lot of studies, and it's important to us that we're really transparent about where we are in that. It's not right for everyone, but there are certainly cases in which this is a great option that provides additional insight to what your next treatment might be. And rather than that communication about our existence as a product happening through social media groups and Facebook chats and things like that, where it's just patients talking to patients, and the information that's traded around might be great and it might not be completely accurate. We felt as an organization that the right thing to do was go to the people that talk to the patients, which are the advocacy groups.
Yep.
And let's make the offer that Cliff spoke to. Let's say, hey, look, we're willing to do this while we're building our data, and we're willing to run it at no cost, as long as the patients we're doing it with understand where we are, understand what it is and what it isn't. And so we want to be the ones that give them that education. And then we also want to be the ones that get connected to their doctor, so we can make sure that their doctor also understands and is comfortable with what's happening.
Absolutely. So let's kind of build a patient profile. So who is kind of the ideal candidate? We've talked about, they've tried a couple therapies, and we've said that it's most cancers, if not all cancers. So who's kind of your ideal profile that this test really has worked for?
Yeah, so the ideal profile is the one that we've sort of described, right? Because of the sort of position of the data as it stands today, which is very positive but still young. It's not typically used in a in a newly diagnosed patient that hasn't had the standard first line therapies yet. The patients that come to us and ask us to work with them and their clinicians are patients that have already been through a line or two of therapy, and they're at that stage where the guidance that the doctor has is educated guidance, but still educated guesswork about what we're going to do next, and their options are becoming more and more limited. And so what we're really doing is we are helping to expand, or actually expand is the backwards word here. There's actually a fair number of options, but it's not really clear on which is best. And so we're helping to narrow that selection process to get them to a drug that might work better for them for that next round of therapy. So it's a patient that generally has advanced disease, and is looking to change therapies soon. Because we're testing real drug response against real live cancer cells, we sort of want them in their current state. Cancer changes, cancer mutates, cancer adapts. Cancer is by definition a mutant, and it keeps doing that. And so, what you ideally want to do is grab the cells at a point that you're looking to make a change. Because if you get the test today and you get some information, and then you do another drug, and then you use the results 4 months later after you've been on another drug, there's a chance that the cancer has changed in that period of time. And so we'd really want to test it before you're looking to make a change. The second real feature that's important is that we do have to have access to live cancer cells. We don't need a lot, but we do need live cells. We need about 100,000 cancer cells to be able to run our full 20 drug panel. Sounds like a lot of cells, but the reality is often in ovarian cancer, for example, and a lot of gynecologic diseases, women in advanced stages will develop ascites, fluid in their abdomen, and that fluid typically is drained at a fairly regular interval in many cases, just because as it builds, it creates discomfort. And that fluid's really not being used for anything else clinically. It's kind of thrown down the drain, for lack of a better word. And so we get a small amount, about 150 milliliters of that fluid that's just being drained as part of your normal standard of care. And it gets dropped into our special test kit and sent overnight, and then we start our process the next day. And then about 2 days after that, we send the report back to the patient's physician. But within those 150 milliliters of ascites fluid, for example, I've seen that small amount contain 4 or 5 million cancer cells. So,
wow.
it can be really effective. And so that's one of the important features, is that the patient needs to have some live cells available. We also can work from core needle biopsies or fine needle aspirations or excisional specimens. So if a patient has a debulking surgery, that's coming up in the near future, or if they're having a biopsy or a fine needle aspiration done for some other clinical indication, we basically just sort of partner with the physician, and we piggyback on those procedures that are already scheduled. Hey, you're going to be doing a core biopsy anyway. Just take an extra core needle for us, drop it into our test kit, and we'll run our test.
Let me make the comment, though, that there's been a lot of activity and development in what we call liquid biopsy, being able to do cancer testing based on a blood draw. And unfortunately, there are not enough live cancer cells in the blood draw for us to run our test. So it does require tissue, not blood, or a malignant fluid, an ascites of the abdominal cavity, or a pleural effusion of the chest cavity, if that happens to be the source of the fluid, or fine needle aspirate, core needle aspirate, surgical excision. So, but some form of getting enough live cancer cells so that we can test them against many drugs.
Sure, absolutely. I think one of the things that amazes me the most about this is how quickly it turns around, and obviously it needs to turn around because the cells will die otherwise, right? So you have to do them while they're alive. But I mean, how many tests have I had that I don't get results for weeks or months, or you're just kind of waiting and waiting and waiting. But this seems like, yeah, you get this done, and then a couple days later you can start the process of making those decisions with your physician and moving forward. It really kind of cuts that waiting process out.
Yeah, that's one of the best aspects of this test, is the 2-day turnaround time. And as you alluded, it has to be a fast test, because otherwise the cells are dead. When you take live cancer cells out of a patient, they tend to die very quickly. And everyone else besides Travera who's tried to do this kind of live cell testing is forced to keep the cells alive for weeks to do testing using cell lines or organoids, or months to do cell testing using mouse models called PDXs. We uniquely have a test, because of this fancy measurement tool invented at MIT, that can do a kind of a 24-hour measurement of the response of cancer cells to cancer drugs. And that enables us to both to return that data to the patient and their oncologist in 2 days, but also it enables us to avoid having to grow the cells to keep them alive long enough to test them. And what's been shown, many, many times and throughout the cancer research literature, is that if you grow up cancer cells, they change. Cancers they're really messed up cells, and trying to keep them alive and having them grow, they don't stay in the current state that they're in, they drift. So after a week, they're still a cancer, but they're a different cancer that came out of the patient. And so testing drugs against that different cancer won't tell you very much about what the patient is going to respond to. So that's one of the really unique and powerful aspects of our test, is just how fast it is, so that we're testing cells that are just like the cells inside the patient.
Well, and so from a layman's perspective, there's really not a downside. Am I missing something?
Well, we do need a live cell biopsy. And so what we invariably do is we piggyback on a biopsy that the patient is already getting for genomics or debulking or staging or whatever it is. So so that's the invasive procedure. But as long as the patient's having that done for another purpose, and Dennis is brilliant at working with the interventional radiologists or whoever's doing the procedure to get some of that tissue draw put into our kit and sent to us overnight. That is the sum total of the burden on the patient.
Wow.
I would say one other thing, though, is, and I want to quantify the comment that Dennis made about we're very early stage in the validation of this. So far, we have run only 55 patients all the way through the process. And by the process, I mean, we get their live cells sent to us overnight via Federal Express. We test a panel of drugs, up to 20 drugs, against those live cells. And then the oncologist gives the patient one of those drugs that we tested. And then months later, we find out, did the patient respond or not respond to that drug? And then we compare essentially our measurement, our prediction of what the patient will respond to the actual response of the patient. And by doing that comparison, we can calculate our predictive accuracy. And our predictive accuracy so far, over the 55 patients that we've tested, is just over 80%. We got 46 of the 55 right. But
Wow.
the catch here is that about a third of those patients who had multiple myeloma, which is our first disease, about a third of them had AML, leukemia, our second disease, and about a third of them have had a solid tumor of some format, which is now our primary focus. But that's a very small amount of data.
Right.
And so the main thing we need to tell patients is we don't yet know exactly what the predictive accuracy of this test is. The early data is fantastic. We'd be thrilled if it stayed this good. But in the absence of having a few hundred patients and at least a few tens of patients in each disease state, and we won't be able to reliably and confidently state what the predictive accuracy of this test really is. And that's why I say we're partnering right now with patients to post, to get them an early version of this test if they'd like to have this, with their oncologist, but also to get us more of that data so we can be more confident that our predictive accuracy is really good.
Sure, absolutely. And the patient profile you're focused on, it seems like they, not to be hopeless, but they might be at their wits' end, right? Like the very last option of, okay, well, what do I have to lose, right? Like, let's do this experimental thing. And it's not necessarily experimental on them, right? I think that's what is so unique because it's a test. Other than the biopsy I'm already going to have or the aspiration I'm already going to have, it really doesn't impact me physically. And so where, the trial and error of drugs might or would actually.
Would.
No, absolutely. That's absolutely right, Alex. And, so kind of there's nothing to lose here, right? If you're already having the fluid drained, if you're already having the biopsy done, send the specimen, let us test it. And, and it's more insight for both you and your oncologists to just make effective decisions from. And it's not like putting an investigational drug in your body that you really don't know the outcomes or side effects of, fully. So it's a little different than some of those other options that exist. But everything that Cliff said is exactly why we're trying to work with foundations and advocacy groups and groups like the two of you, is to get the right education out there, to make sure that patients understand what this is and where it is, and have those honest conversations with their doctors.
Yeah. Just out of curiosity, how have you seen the response from the oncologists who receive the report? Do you find that 100% of the time they take your recommendation, or what is that something you can share?
Sure. No, that's a great question. We sort of spoke to or alluded to earlier that that this is new and some doctors are going to be apprehensive. And part of the reason they're apprehensive is, we also noted that we've been trying this kind of this sort of ex vivo drug test approach in cancer for a long time. And in fact, since the 1950s.
Wow.
So there's 70 years of experience, and sort of failed experiments, for lack of a better word, of this kind of an approach. So a lot of oncologists are appropriately, apprehensive and kind of doubt what it is we're trying to do here. So what's always really important in my communication with patients is that as they go into their conversation with their oncologist, they make it clear that they understand that. Look, we understand that this has been tried and failed. Here's what's different. Every previous generation approach that's happened over the last 70 years has had to grow the cells. And growing them changes them. And so this is the first time someone has had a clinically applicable assay that tests the patient's direct cells for that drug response. And so that is really what makes this special and unique. And when we have the opportunity to either educate the patient that they can take that to their doctor, or that we can work with and educate their doctor directly, physicians that understand how different this is We're doing it in sort of a trial basis. There's no financial risk. There's no other risk beyond procedures that are already being scheduled and done for other clinical reasons. And it's just additional information that if the doc is really uncomfortable with at the end of the day, if the assay result made a recommendation of a drug that the doctor's like, this is crazy, there's no way I would give this drug. Most patients are going to say, okay, I get it, I understand. But usually the kinds of recommendations we're making are in line with drugs that they already are considering. And so the response of physicians that have had a late stage patient bring this to them and ask for it has been really very positive. Sometimes some convincing needs to occur, and some education needs to occur, but generally they go, you know what, we've tried a lot of different things. Our options for you, frankly, are running out. And so, you know what, it doesn't hurt. Let's get the test done. Let's see what it shows us, and then let's make a decision. So the vast majority of physicians that have had patients bring it to them are doing it. And many of them, I think, are using the results. But that's kind of that data that Cliff spoke to earlier of those 55 patients that we have full outcomes on. We've now tested, significantly more than that. I mean, we're probably in the 300 range of total cases, somewhere near that. And for many of those, we're waiting to collect that data, on the back end to find out, did you go on one of the drugs we analyzed? And if so, was there a response? And so we don't have a lot of that information yet, but we're continuing to build that.
And so not only is there not a risk, but there's every reason to do it, not only for the patient, but for future patients once you get that data.
Yeah, we're very thankful to the current patients who are, willing to take their time and energy and learn about it and talk to their oncologist. That's the cost to them. But because even if it turns out that their oncologist chooses not to use our advice, there's value in us as we're learning how to do the test better. Often, sometimes we actually end up in a negative situation where we predict a drug won't work, but the oncologist says, no, I just used this for 5 patients. I know this is going to work for the patient. You give it to the patient, and then we get to evaluate, hey, can we call negatives as well? Can we say, don't use this drug because we have evidence that it won't work? Of those 55 patients that we tested, about half of them were these negative tests where we tested a drug and said, we don't think this one's going to work. These were just in an observational study, so we weren't telling the oncologist what to do in any way. And the oncologist was like, no, that's my favorite drug, I'm going to go use it. And they used it and it didn't work. And of all of those cases, we got 80% of those right too.
Wow.
So it might be an enormous way to avoid toxic drugs that aren't going to do anything.
Right. Well, and I'm sorry, go ahead.
I say the other really interesting use case that came up very early in our history was there patients who go on to a drug, typically, maybe a very popular chemotherapy, a cisplatin or a carboplatin or paclitaxel or one of those, and it works, but it works for a while, then they fail the drug. And then they go on to something else. Well, later in their course of treatment, we've talked to oncologists who said, I have a suspicion that cisplatin would work again for this patient, given how their cancer has evolved. But it's a very hard thing to do, to ask a patient to go back onto a toxic drug that they've already failed previously. Well, we can run the tests on all the drugs that they failed previously and measure, we're putting molecules of that drug right onto the surfaces of those patients' cells in our laboratory and determining whether or not those cells are responsive to that drug. So when we get a response in our lab, that provides both the oncologist with guidance like, aha, this cancer has changed, it's kind of come around again. We can use a tried and true drug and it'll have a positive effect, and gives the patient some comfort that they're not going on to a toxic drug for a second time when it failed the first time, but there's a really good reason to give it another chance. So there's some wonderful cases like that where we're not, going outside the bounds of the things that should be tried for that patient and that cancer, but kind of provide insights to the oncologist that they otherwise couldn't get without such a test.
Sure.
Another question, just out of curiosity, when this evolved and turned into what it is, this measurement tool, is that what MIT set out to do, or was it one of those
It's a great question,
happy
man.
accidents?
No, come on. It's MIT. I'm an MIT alumnus and I love the place dearly. And they do an awful lot of technology for technology's sake. They build a cool new thing and then show it works, and then you kind of lift your head up and look around and go, now what do we do? And in fact, the laboratory that made this invention is the Manalis lab at MIT. Used this invention, this cell weighing tool, for all sorts of basic cell biology research.
Wow.
Before they said, we talking to my buddy over at the Dana-Farber Cancer Institute and had a really cool idea about maybe this could be used to solve this unsolved problem of figuring out which cancer drugs are going to work for which cancer patients. And then, years of research ensued before we finally got to the point where we say, yeah, this is far enough along where it's actually worth bringing it into commercial practice.
Well, and, isn't that the beauty of creativity?
Yeah, indeed it is.
Especially in the scientific realm.
Yeah, I mean, the great academic institutions, not just MIT, but the 50 or 100, depending how you count, research institutions in the United States. These represent the greatest creative discovery platform in the world. And I'm very, very proud to be affiliated with MIT and do everything I can to support that kind of ongoing, basic research that leads to applied research that leads to improvements in medicine and other fields of science as well.
Sure. So where do you see this going in the future, or where do you hope it goes in the future? And what's your timeline looking like of when is this going to be in every doctor's office, a regularly suggested test?
Well, and related, I was just going to ask about distribution and marketing. Are you already looking down the road at that?
Yeah, our timeline right now is slow and cautious because the number one thing we need to do is establish our clinical validation data set. And so we're keeping this very small, very closed, carefully qualifying the patients and the oncologists we're working with, because we don't want it to get out of control and get misused in any way until we sort of fully understand how to use it. And we'll be doing that for another 1 to 2 years. At that point, we'll be ready to really start scaling it up. We'll expand our facility, we'll expand our patient profile, we'll expand the number of samples that we run, and ultimately generate enough data to go to Medicare, to go to CMS, and then the other private insurers, the Uniteds and the Aetnas and places like that, and say, hey, look what we can do. We can improve patient care. We can change the probability that next treatment has from a 20% likelihood of it working to an 80% likelihood of it working, which is terrific for obviously for patient care. Also to the payers, as you can well imagine, we can say, hey, we can identify which drugs aren't going to work.
Right.
That's a big deal because everybody knows more than half of all cancer treatments don't do anything for the patient. We just don't know which half, right? So that's the next step. And then we will build out a very large laboratory. We don't quite know where yet. We have a lot of flexibility, but a very large laboratory, not necessarily. Our current laboratory is in Boston. We're still kind of connected at the hip to MIT.
Sure.
But that won't need to be true in the future. And then we can scale it up around the United States. But there's a bigger plan. And the bigger plan is, this is a very small and relatively inexpensive benchtop instrument. There's nothing fancy about it. It has a very fancy core, the chip that actually does the measurement technology itself. But after that, it's a very common benchtop instrument like you'd find in any diagnostics lab. And so we plan to package those instruments up for users that aren't just us in our laboratory, but for anybody to use it. Take it through the FDA process of getting it as approved as an in vitro diagnostic device, an IVD. And then we can sell instruments and kits, meaning kits containing drugs for each individual cancer, to every cancer hospital in the world.
Oh, wow.
Wow.
And then the 2-day turnaround time goes to 1 day.
Yeah.
Because
Yeah.
Right.
you no longer
You don't
have
just send it
FedEx.
off.
Yeah,
Right.
no FedEx in there. And so a same day therapy selection test that applies to virtually all cancer patients and virtually all cancer drugs is really the vision of the company.
Yeah, then that was my next question about FDA approval or any kind of licensing or anything you have to go through.
Yeah, we're on that long path to get to FDA approval, but through CLIA certification, which we are now, we're able to provide oncologists with the results of these tests.
Yeah, and that's an important note, actually, Mary. And as Cliff just said, with this is classified as an LDT, or a laboratory developed test. And what essentially that means is that the laboratory that created the test and created the technology is the same laboratory that's running the test.
Okay.
And so and there's a lot of tests out there like that, tests like Oncotype DX or MammaPrint in the breast cancer world that a lot of people have heard of. Those are LDTs. There's numerous examples. BRCA testing, when it started, really started as an LDT. So these kinds of assays don't have to be FDA approved or FDA cleared. They just have to be CLIA approved and run through the CLIA labs that developed them. And and so that does give some opportunity for us to do this a little differently. But that CLIA approval does require a physician's order to be able to run. And so that's why, as we work with patients and as patients find us, the first thing I do is I communicate with them, is work with them to get connected to their clinician so that we can, get their doctor's buy-in and make sure that everybody's on board with what's happening. The FDA piece, it really comes in when we start selling that box, like
Right.
Cliff was talking about. The other thing that I'll add to the other comments Cliff made a moment ago that is, I think long-term vision for me, at least, coming in as as pretty much the new guy in the organization, is right now, with the level of data we have, this assay has some really important applications in late stage disease. But the reality is that there's oodles of data out there to show that patients with earlier stage disease on first lines of therapy, even neoadjuvant approaches when they get therapy prior to surgery to try to shrink the tumor for better surgical outcomes, all of those approaches, the better initial response the patients have in a lot of those cases often translates to better long-term outcomes as well. And so being able to select the right drug up front, even in a newly diagnosed early stage patient with a test that can turn around that biopsy in 2 days, could be really valuable. But to get that kind of an application in early stage disease, the level of data, the volume of cases, et cetera, needs to be much, much higher than what it is in later stage disease. And so that's a long-term view, but I think as we get to the place where there are cancer centers around the world that are running this, I could really foresee those cancer centers not just running this for late stage cases, but potentially for all cases.
Wow. I mean, that's a game changer. Big time.
It's a big vision. It'll take a couple days to get there.
Hey, and that's okay. That's what it's all about, looking ahead.
Absolutely. And so just from a technical standpoint, I wish I'd asked this earlier, but so can the microchip be used over and over, or is it for each test there's one microchip?
Yeah,
so that they would have to like replace it.
I'll even give you 90 seconds of physics at your peril here. But yes, these are what we're called, the microchip is called a MEMS chip, a microelectrical mechanical device. It's made in a semiconductor fab, and we're currently using a very high-end fab in France to make these chips, although we've done them in Southern California as well. And the chip is reusable. They don't cost a lot to make, and they are reusable, so the cost of the test is very manageable. But here's how it actually works, in case you have readers and viewers who'd like to understand it. Inside this chip, which is a fluidics device, you can pump liquids through it. Inside this chip are 30 micron channels. So that, and 30 microns is about the diameter of a single cell. So you can pump single cells through this chip. But in part of the chip, there's what we call an oscillating cantilever. This is a very fancy physics term for a diving board. Diving board that vibrates up and down. But it's a very special diving board, because inside that diving board is that 30-micron fluidics channel that runs down and back the diving board. And outside the diving board is wrapped with vacuum to let it oscillate freely. And we pump single cells through this diving board. And we take advantage of a very simple law of physics that says the natural resonant frequency of a diving board, of an oscillating cantilever, is directly proportional to its mass. So that when you pump a single cell through the cantilever, its resonant frequency changes by a tiny amount, by essentially the amount that is the weight of the cell. Well, measuring frequency change turns out to be one of the most accurate measurements in all of physics. You can measure a frequency change to about 10 parts in a billion with a $100 FM circuit. It's cheap and it's super accurate. So that accurate measurement of the frequency change of the oscillating cantilever directly translates into an incredibly accurate measurement of the weight of the cell. And then so we run these systems all day, and at night we shut them off. In the morning we wake up, we run bleach through the system to clean the whole thing out, then run fresh water through it, and it's ready to go for the next day.
Wow.
So it's like I say, it's a very physics-oriented solution to what people have been trying to solve with biochemistry for so many years. And biochemistry is so hard, and the physics of semiconductors is so well understood that we're able to take full advantage of the physics that was invented at MIT, and really been the core technology we've used.
Yeah. Well, and how incredible that someone said, wait, I have a friend in this lab and they might be able to, or just a conversation over coffee of, yeah, we have this new invention, and, how incredible.
Welcome to the world of scientific invention.
Yeah.
That's just how it works. That's how COVID was so horrible. We couldn't stand around the water cooler anymore.
Right. Exactly.
And we're all back, thank goodness. But it was definitely a pause there. So.
Well, and I'll add a little bit to Cliff's description just now, because I'm the guy that doesn't have a degree in physics from MIT. So, Mary, when you were like just 4 or 5 years younger, and Alex was like 7 or 8 years old, and the two of you would go to the swimming pool, and Mary would jump off of the diving board. The diving board vibrates after you jump off of it, right? And then Alex would jump off the same diving board, and it would vibrate too. But the vibration is different based on how much you weigh.
Right.
And it turns out that continues to be true all the way down to really, really tiny differences.
Wow.
And so that's, at the end of the day, what this is. It's measuring the vibration of the diving board as these cells pass over the top of it.
Well, and that simple explanation or comparison really must help in explaining it to patients. To not get all scientific with people who aren't into that, but to help them understand.
Absolutely. And to doctors too.
Yeah.
And all we're doing is we're taking advantage of an incredibly well-known fact that dead cells weigh about half what live cells weigh. Okay. So you should be able to weigh the cells and figure out, if they're on the path to death. Now, if you wait all the way to the time they're dead, it's too late. You'll never figure out, was it the drug that killed the cell, or did the cell die a natural death? That's the problem. But because our instrument is so sensitive, we're not measuring that half of the weight is lost, all the way to the end of cell death. We're measuring the very beginnings of cell death. We're measuring that teeny little weight change that indicates this cell is going to weigh half of what it weighs right now, but it's going to take it a week to get there, and we can make this measurement in a few hours.
Wow.
And that's the secret sauce here. I always say, all we really have is speed. We have speed because we have sensitivity. That is the speed that gives us the accuracy. So, this is a measurement tool, and what is DNA sequencing? It's a measurement tool. I'm a measurement tools kind of guy, and measurement tools change the world. It's like, I would say, what is the scientific method? Hypothesize and test. hypothesizes having a bunch of cool ideas. Testing is about measurement tools.
Mhm.
So whenever we come up with a new measurement tool, science moves forward. And one of the key sciences that moves forward is medicine. And that's exactly what's happened here. A new measurement tool from MIT, plus a partnership with Dana-Farber to identify the right application for it, has enabled this measurement tool to move the science of medicine forward in a way I think is going to be very helpful for cancer patients.
Yeah. Well, and you said it, speed is incredibly important, particularly in cancer, particularly in late stage cancer.
Yep.
Every day counts in decision making.
Absolutely.
It does, yeah, and yeah, that's been one of the biggest pieces of feedback that we've gotten. But what's really cool as somebody who has spent 15 years in genetic and genomic testing world, what's really cool about this test, you guys have both sort of hit on, it's that this is an incredibly complex, scientifically heavy assay at its core, but it just kind of makes sense.
Yes.
From a logical perspective, it's pretty simple.
Yep.
And when I speak to patients and sort of explain, the diving board or however we go about explaining it, patients just go, so you're going to put drug on my cells, and then you're going to tell me if their weight changes in response to the drug? That makes a lot of sense. That's really
Like,
cool.
I get it.
Yeah.
Right.
Yeah.
Yeah,
It's
I'm not talking to him
fascinating.
about SNPs and splits and mutations of this and that. It's an incredibly cool, very functional approach
Yeah.
to finding out what's going to work in a specific disease.
Yeah. That's incredible. So if someone is listening today who thinks that they fit the profile, they are a current cancer patient, they are ready to change their treatment, their doctors are talking to them about being in late stage, or they've tried a couple treatments, how do they get in contact with you? What is the next approach, or what's the next step?
Yeah, no, great question. So I would recommend a couple of different things. The first is you can go to our website, www.travera.com. It's Travera. And there's a contact us form on there that if you fill out, that email actually comes directly to me. And, we're a small group, we're a small team right now. And so I'm the one that actually works with every single patient that comes through our door. The other option is just to come directly to me. And if you don't want to go to the website, and I'll give my email address is dwatson@travera.com, and we'll provide that for you guys to put on the screen. But just reaching out to me directly to initiate the process. And then generally what I do when I first hear from a patient, whether I get a phone call to our lab or to my personal cell phone or through email, I have just a standard email that I start with. Once I connect with them, that just says, look, this is the process. I remind them again of where we are in the data. I remind them again of some of the sort of important features and facts. You've got to get your doctor's support. You've got to get your doctor's buy-in. They have to order it. You're going to have to get fresh tissue. We can't work from old frozen specimens or archived specimens. And just sort of line out all of the steps, and then even provide some guidance with how they can initiate the conversation with their physician. And so, because of PHI and HIPAA, I can't reach out to Alex's doctor without Alex first making that connection.
Right.
I can send them an email, but they're not even going to acknowledge that you're one of their patients, right?
Right,
So I always have the patient initiate that contact, and then I try to pick up the ball from there as much as I can. Once we get the doctor involved and in support, what often happens is we actually send the test kit to the patient. So the patient takes the kit with them the day of their procedure. And in some cases, even after the procedure, the doctor will return the test kit to the patient so the patient can make sure it gets out to a FedEx site that day. Some hospitals will just put it with their outgoing stuff. Some want to hand it back to the patient, but it becomes a pretty simple process. And we just kind of work through it, and whatever is the unique variables of each person's situation, but we help line out all of those details and try to make it as simple as we can.
That's fantastic. Thank you. And yes, we'll put all this information on the screen on our website, so that anyone can, who's interested in learning more, can find out and get in touch with you guys. Because, for patients who fit this profile of what you're looking for, it's just an invaluable tool to help them, figure out their next steps.
Well, and let me just say, for you to be willing to take a Friday afternoon and talk to us so that the word gets out, I think is going to be the heart of your success in getting the word out, because you're willing to do it and take the time. And that means a lot, because they're hearing it from the people who know it the best.
Mm-hmm.
Absolutely. Well,
And
thank you for giving us the opportunity to do this. We're all in this thing together. We're all trying to get the best, treatments to all patients, but in this case, more late stage patients that we possibly can. And we think we've got a tiger by the tail here. We think we've got a new way of doing it that is really going to be valuable to the patient community.
Yeah, absolutely. Well, thank you so much for joining us. And like I said, all this information will be on our website. And we hope that we will have an update later on of how wonderful Travera and this test are doing, because you guys are doing some really great work. We really appreciate you.
Let's keep in touch. And
absolutely.
We'll let you know. So
fantastic.
Yeah,
Thank you
both. This has
both.
been a lot of fun.
All right.
Thank you guys.
Yeah.
Thanks so much.
Thank you.
Thanks
so much.
Bye. Thank you.
Bye.
Thanks for listening to this week's episode of Down There Aware. Be sure to like and subscribe on our new YouTube channel, as well as on your favorite podcast platform. You can find us at Down There Aware on all social media sites: Twitter, Pinterest, TikTok, Facebook, Instagram. If you have it, we're there at Down There Aware. You can learn more on our website, www.downthereaware.com, and always get in touch with us, downthereaware@gmail.com. See you next week.